All right, now that you know who Gregor Mendel was and what he did and what he discovered, it's time to go beyond Mendel and look at what's been discovered since his discoveries. And one thing is this concept or this phenomenon known as incomplete dominance, where one allele does not completely dominate over the other allele. As we've been looking at everything that Mendel looked at, all 7 characters that Mendel investigated were completely dominant, either completely tall or completely short, completely round or completely wrinkled, and so forth. In incomplete dominance, the intermediate, the heterozygote, shows a phenotype that's intermediate between the 2 homozygous conditions. So there's an intermediate phenotype, which the effect of the individual alleles cannot be distinguished. And that's important because there's another concept we're going to be looking at on the next slide known as codominance. And in codominance, the individual alleles, the effect of the individual alleles can be distinguished. So this is an important part of understanding the difference between incomplete dominance and codominance. In incomplete dominance, you can't look at these pink flowers, for example, and see the effect of the red and the effect of the white individually and distinguish them individually. They just kind of blend together to produce pink. So if this was going on in the 7 characters that Mendel investigated in pea plants, it would have really confused him, and he may have not as easily discovered that heredity is by particles or genes, because this kind of supports the blending hypothesis. So this example is what are known as 4 o'clock flowers, and they have incomplete dominance in their flower color, where the heterozygotes are pink. And Mendel was lucky, like I said, that none of his 7 characters were inherited this way, because it really would have confused him. This is still the Mendelian procedure, though. We're starting with pure parents, pure red and pure white, and the offspring are all heterozygous in the F1 generation. And now let's take a look at what happens if we go on from the F1 generation to the F2 generation using a Punnett square. So F1 to F2, both parents are going to be heterozygotes, and we end up with what we with the same results that we usually end up with in the F2 generation in Mendel's experimentation, where we have one that's homozygous dominant, homozygous red in this case, one that's homozygous white, but then the heterozygotes are going to be pink. So as usual in the F2 generation, the genotypic ratio is 1 to 2 to 1. 1 homozygous red, 1 homozygous white, and 2 heterozygous pink. Always in this order, though. Homozygous, well, in this case, homozygous red, heterozygous, homozygous white, 1 to 2 to 1. And if you look at the phenotypic ratio, it's the same thing. 1 red, 2 pink, 1 white, 1 to 2 to 1. So if you're ever dealing with a genetics question on a test, for example, and the phenotypic ratio is the same as the genotypic ratio, that's a clue that you're dealing with incomplete dominance. Also, if they show you symbols like this, in other words, there are no lowercase letters representing recessive alleles, that's another indication that you may be dealing with incomplete dominance. All right, as promised, here we're looking at codominance, and we're looking at 2 examples in chickens and in the ABO blood grouping. You probably are familiar with the fact that you might have either type A blood, type B blood, type AB blood, or type O blood. And it turns out that type A and type B are what we call codominant. And in codominance, as opposed to incomplete dominance, the effect of the alleles are fully distinguishable in the phenotype. So the phenotypic effect of the alleles are fully distinguishable. It's easiest to understand here with the chicken example. That's why I included it. So In this chicken, the feather color is the result of two alleles, one for white and one for red. One for black. So we end up with some feathers being white and some feathers being black, and you can very clearly see that some feathers are white and some feathers are black. If this was incomplete dominance, then this chicken would be gray, and you wouldn't be able to distinguish the individual feathers that were either white or black. But you can, and that's because they are codominant. Neither one is dominant over the other, and they're both expressed. And you can distinguish their expression individually in the phenotype of the individual that inherits both of them. We see the same thing here in blood type. What's being shown here are red blood cells, and what produces different blood types are proteins that are on the surface of red blood cells. In other words, in the plasma membrane of red blood cells. And here we see what we call the A antigen. These proteins are referred to as antigens because an antigen is something that will cause an immune reaction. And that's why we're concerned about these proteins on the surface of red blood cells, because if you put blood, like type A blood, for example, into a person that has type B blood, then their immune system is going to see these A antigens, these A proteins, as being a foreign invader and attack them, and therefore destroy the red blood cells. So people that have type A blood have the A antigen on their red blood cells. Those that have type B blood have the B antigen. Those that have type AB blood have both, and that's the situation that describes or is an example of codominance. Both A and B are codominant. And so a person that receives both the A and the B allele, notice that they're being represented here by capital I's. Both of them have capital I's, and then a superscript, capital A, superscript, capital B. Everything's capital because it's codominant. Nothing is recessive. And a person that has type AB blood, it's because they've received from their parents both the A and the B allele, and there are both A and B proteins on their red blood cells, and those are distinguishable by doing a test, a blood type test, but they are distinguishable on the red blood cells. And that's what makes this codominance, not incomplete dominance. And we're going to be looking at blood type again when we study human genetics and biotechnology, that unit. But just so you hopefully understand what's going on here in blood type and what makes these different blood types different, and why you can't give them to just give any kind of blood to anybody. Again, that's the A antigen, which, if put into a person with type B blood, will be seen as a foreign invader, and the immune system will attack them. So an antigen is any molecule, and it's usually a protein or a piece of a pathogen that will produce an immune response. We came across this word back when we were studying bacteria and viruses too, because a vaccine, usually what you're injecting, or not usually, always what you're injecting to vaccinate someone against a virus or a bacteria is a part of that virus or a part of that bacteria, an antigen that will produce an immune response. So an antigen is anything that will produce an immune response. So on type A blood, there's the A antigen. On type B blood, there's the B antigen. On type AB blood, there are both A and B antigens. And on blood type O, there are no antigens. We're also looking at multiple alleles here in ABO blood type. So that's another characteristic or type of inheritance or heredity that goes beyond what Mendel knew about. Multiple alleles, meaning that there are three alleles for blood type. There's the A allele, there's the B allele, and there's the O allele, and the O allele is recessive to both A and B. So A and B are codominant, and O is recessive to both A and B. But the idea in multiple alleles is that out in the human population, in the human gene pool, in all the humans that are out there on the planet, there are three alleles that code for blood type, but each individual can only have two. because you get one from your mother and one from your father. So each individual only has two. If you look at all of these genotypes, there are only two alleles. So there are two ways to have type A blood. You can be either homozygous A or heterozygous A. But there are only two alleles. In either of these genotypes, there are only two alleles. You can be homozygous B or heterozygous B. And you can be heterozygous AB, codominance, and then to have type O blood, you have to be homozygous recessive, just like always. But there are these three alleles in the human gene pool that produce type A, B, and O blood. And just to emphasize the different genotypes here, I went and underlined them and forgot that I underlined them. I should have pulled them up as I was explaining. So there are three alleles. That's what makes it multiple alleles. A, B, and O. And we have to use different symbols. So again, if you see symbols like this, it's an indication that you're dealing with multiple alleles. Just like on the next slide is another example of multiple alleles. So again, no antigens, homozygous recessive in type O blood. Here's, like I said, another example of multiple alleles. In the case of coat color in rabbits, this certain species of rabbit anyway, there are 4 alleles in the gene pool of the rabbits. But notice that when you look at the genotypes, there are only two alleles for each genotype. So again, even though there are 4 alleles in the gene pool of the rabbits, the population of rabbits on the planet for this species, that doesn't mean that a rabbit can receive more than 2 alleles. So it's just another example of multiple alleles, but even more than 3 in this case. And what's interesting here is what I call the hierarchy of dominance, where full color, which is what we're seeing here, is dominant over the other 3 alleles. And then chinchilla, which is this coat color, which is the same color as chinchillas, if you know the natural coloration of chinchillas. That is recessive to full color, but it's dominant over Himalayan and albino. Himalayan, which is this one, is recessive to both full color and chinchilla, but dominant over albino, and then albino is recessive to all of them, the other 3. So in the interactions of multiple alleles, things can get somewhat complicated. And before we leave this concept, I just want to remind you that alleles are different versions of the same gene. So these all originated by mutation, and they stayed in the population because they were selected for by natural selection. That's how we end up with more and more and more alleles of the same gene. All right, not to be confused with multiple alleles is a concept known as polygenic traits. And that is one that results from the expression of more than one gene. And we're looking at skin color here as our example, human skin color. And this is the opposite of a single gene trait. All the 7 characters that Mendel looked at in pea plants were single gene traits. And the characteristics of a single gene trait is either or, either tall or short, either round or wrinkled. There are only 2 possible phenotypes. But in polygenic traits, there's a range of phenotypes. So again, all 7 characters that Mendel studied were single gene traits, so he was lucky that way. Polygenic traits are most prevalent in the human genome. In other words, most human traits are polygenic traits. Skin color, again, being an example. So this figure is only showing 3 genes, A, B, and C, but human skin color is actually coded for by at least 7 genes, the interaction of at least 7 genes, and probably more. We just haven't discovered all of them yet. And it's the same for height and weight and intelligence and hair color and eye color and so forth. All of them are, most human genes or human traits are polygenic traits. So again, if you look at the diagram and how skin color is inherited, you'll notice that the most extreme light shade of skin has all recessive alleles, and the most extreme dark shade of skin has all dominant alleles, and then there's everything in between. So the idea is, as you go from very light to very dark, there's the addition of dominant alleles. And so that's referred to as a quantitative effect or an additive effect. The quantity of dominant alleles is responsible for a darker skin color, or the addition of dominant alleles results in a darker skin color. So again, you end up with this spectrum or range that you don't see in a single gene trait. A single gene trait, there would be only 2 bars on, if you bar graph it, there would be only 2 bars, like 1 bar for tall and 1 bar for short. So this is a histogram, and a histogram is a graph that shows the distribution of a variable in a population. So that's what we're looking at here, is a distribution of skin color in the human population. And it's not complete, it's just an example, because again, there's really more variation than what's being shown here, because we're only looking at 3 genes here as an example, but there are actually over 7. But this is referred to as normal distribution. Whenever you see a bell-shaped curve like this, that's referred to as normal distribution. So a bell curve, and it indicates a polygenic trait. So if you ever see a graph on a test, for example, that shows a bell curve, that shows a range of variation for any particular trait, then you know you're dealing with a polygenic trait, because there are a range of phenotypes. It's the opposite of either or, like either tall or short or round or wrinkled. So again, this polygenic inheritance goes beyond what Mendel discovered, and so does the work of Thomas Hunt Morgan. So this is Thomas Hunt Morgan, experimented in the early 1900s with fruit flies, because again, just like pea plants, fruit flies are great for genetic research. So what he did was he and his students looked at thousands and thousands of fruit flies looking for mutations, and what he was doing in his work with fruit flies, or what he ended up doing in his work with fruit flies, is confirming Mendel's principles. The idea that Mendel's principles don't just apply to pea plants, they apply to every living thing on the planet, all organisms, including, fruit flies. So not just plants, then animals, and all species. So again, they search for mutations. The mutation shown here is white eyes. Usually fruit flies have red eyes. I don't know if you've taken a close look at fruit flies, but they all have red eyes. But occasionally, and like I said, they had to look at hundreds of thousands of flies before they finally found this white-eyed mutation. But when they found this mutation, they then used that individual to breed in breeding experiments to figure out how it was inherited. And so this describes his experimentation. So when Morgan crossed a pure and red-eyed female, pure red-eyed female with a pure white-eyed male, because the one that they found was male, and you'll see why in a minute. And this is a normal Mendelian experiment. So Morgan's following Mendel's experimental design here. So we're crossing a pure red-eyed female with a pure white-eyed male. All the offspring in the F1 generation had red eyes, which is expected if red is dominant over white, right? So nothing new there, nothing unexpected there. When he crossed the F1 hybrids, he observed a 3-to-1 phenotypic ratio in the F2 offspring, which is also expected. That's the same thing that Mendel always saw, 3-to-1 phenotypic ratio. What was unexpected, though, is when he took a closer look, he realized that all of the white-eyed flies were male, just like the one that he had found in the first place. All the white-eyed flies were male. And he was able to conclude from that, figure out what was going on, is that eye color is a sex-linked trait. Just like in humans, hemophilia is a sex-linked trait, color blindness is a sex-linked trait. And what that means is that the gene for that character is on the X chromosome. So a sex-linked trait is a trait in which the gene is on the X chromosome. And just a little review about gender determination or sex determination in humans and in fruit flies. Female is 2 X chromosomes. If you have 2 X chromosomes, that results in a female. If you have 1 X chromosome and 1 Y chromosome, you end up being a male. So that's the X chromosome we're talking about. We're talking about these sex chromosomes that determine gender in humans and in fruit flies, and it works the same way in fruit flies as it does in humans. So the significance of Morgan's discovery here was that he was the first to demonstrate experimentally that genes are on chromosomes. People have been looking under the microscope at how chromosomes divide up during cell division and gamete formation, but no one ever was able to show experimentally that the genes that are responsible for heredity are actually on the chromosomes. So this was the first experimental evidence that genes are on chromosomes, and specifically in this case on the X chromosome. So he discovered basically 2 things. Genes are on chromosomes, and he discovered this form of inheritance that we know as sex linkage, or form of heredity we know as sex linkage, sex-linked traits. And sex-linked traits end up being more prevalent in males than in females, and we'll take a look later at the genetics of this using Punnett squares. So unfortunately, humans are not good subjects for doing genetic experimentation. You can't ask 2 people to mate with each other, select 2 people to mate with each other to produce offspring and analyze their offspring. That just doesn't work with people. So it's a good thing that we have some good experimental subjects to work with, like fruit flies. Human heredity can be studied, however, by looking at pedigrees. In other words, looking at the family history. And so it's good to know, like, for example, if your great-great-grandparents had a particular genetic disorder or something, then these scientists known as genetic counselors can look back at your family history, at your pedigree, and figure out what you are likely to be affected by. So there are more tools that genetic counselors can use besides just pedigrees. They can also use genetic testing, and that's becoming a big one now. So even if they don't know your family history and can't really put together a complete pedigree for your family, they can do genetic testing to see what genes you carry. They can actually test for particular alleles. So as I've referred to here, this is a genetic counselor, and you can see what's all over the walls are pedigrees. So the pedigree above here indicates a sex-linked recessive trait. And your clue is that only males fully have that trait. Females can carry it, but they don't show that trait. Only males are showing the trait, and that's your clue that you're dealing with a sex-linked trait. So we can assign genotypes. Once we realize it's a sex-linked trait, then we can assign genotypes to the individuals here. And what I'm asking about specifically is the probability that a female in the lower right, so that's this one, will have an affected children if she mates with a normal male. So if we assign genotypes here, and we know this male has to be, and so notice the symbols that we use when we're dealing with sex linkage. Again, the gene's only on the X chromosome, so there is no gene on the Y chromosome. There is no allele on the Y chromosome. So the allele is only on the X chromosome. And a male we're looking at a recessive trait here, and we're using hemophilia as our example. So that's why I chose H. So normal is dominant over the recessive hemophilia, right? So we use a capital H, superscript capital H on the X chromosome. So a male is going to have the dominant allele, and there is no other allele on the Y chromosome. We call this hemizygous. So the male here is hemizygous normal. And then the female is a carrier, so she's heterozygous, and she can be heterozygous. Only females can be heterozygous when it comes to sex-linked traits. because they have two X chromosomes, and therefore they can have two alleles. So this female is a carrier. A heterozygous female is going to be a carrier of a sex-linked recessive trait. Then this female, same thing. This male, same as this. So any normal male is going to have that genotype, and any carrier female is going to have this genotype. And again, we're just assigning genotypes to those individuals that are involved in tracing down the family history of this female so that we can figure out what she is. Here we have a male that's affected, therefore he is hemizygous for the recessive allele. In other words, lowercase h on the X chromosome that he received from his mother, this mother. And so this female then is a carrier, and the genetic counselor would be able to figure that out by looking at the family history. And just to complete this, her brother is normal. But we're interested in what's going to happen if she mates with a normal male, so we have to do a Punnett square to figure that out. So here's the male, the normal male, right? Same genotype as a normal male. And then here is her, the carrier, female. And if we then complete the Punnett square, the question is which of her children would be affected by hemophilia? Well, the only one that would actually have hemophilia is a son, is a possible son. Remember, what's here is possible, not for sure, but possible. But she could also have a son, or they could also have a son that is normal. So it's predicted that half the males would would have hemophilia, and it's predicted that half the females would carry hemophilia. So if you consider carrying to be affected, then that means that there's a 50% chance that their children would be affected by hemophilia, either have it or carry it. So we're still going on beyond Mendel here, and Mendel really had no idea of the effect of the environment on the phenotype of offspring. So here we're looking at the question that's often referred to as nature versus nurture. How much of us is coded in our genes, and how much of us is the effect of the environment or our upbringing? That's the question of nature versus nurture. And the idea in this figure here is that these hydrangea are genetically identical. Right? They're both hydrangea, and supposedly they are genetically identical. And this one is in acidic soil, and this one's in alkaline soil, and hopefully you remember that alkaline refers to basic, so soil that has a high pH, acidic being soil that has a low pH, and that accounts for the difference in their phenotype, in their color. So if you grow hydrangea in acidic soil, they're going to be bluish, and if you grow them in alkaline soil or basic soil, they're going to be more pinkish. So no matter what their nature, meaning their genetics, their nurture, their environment that nurtured them has caused them to be different from each other. So again, nature versus nurture refers to how phenotype results from the multifactorial interaction. I know that's a big word, but multifactorial, just more than just genetics, right? Both genetics and the environment are factors that are working together and interacting with each other to produce the phenotype that we see in the individuals. So it's multifactorial. Genes, nature refers to genes, and nurture is referring to environmental factors, like the soil pH in this case. So this arrow diagram is representing the same thing. Genetic, meaning nature, on the left, and nurture, being environmental, on the right. And I've thrown in a couple other examples here. So like dwarfism is a genetic disorder, purely genetic. It really doesn't matter what the environment is. If you receive the genes or the alleles responsible for dwarfism, you're going to be a dwarf, no doubt about it. So these things that are basically genetic disorders, that means if you carry the alleles for those disorders, you're going to have those disorders. At the other extreme, though, tuberculosis, scurvy, diseases in general that are caused by bacteria and viruses, and also things that you may be exposed to, like mutagens, carcinogens, like those contained in cigarette smoke. So you're much more likely to develop those things if you are exposed to the nurture, the environmental factors that cause them. And then we have in between multifactorial things like diabetes and ulcers and heart disease, obesity. So you may be predisposed to these things genetically, but you're not necessarily going to have those phenotypes if you do certain things or are exposed to certain things or not exposed to certain things. So, for example, you may be genetically predisposed to obesity, but if you work out like crazy and watch your diet, you're not necessarily going to be obese. What have been very useful in these kind of studies are identical twins that have been separated at birth. So they've been studied, and it's been useful to show what in humans is genetic and what in humans is environmental, that produce the way that we are. And then there's a whole new level of genetics that really goes way beyond Mendel, known as epigenetics. And it's been discovered that there are heritable environmental modifications of genes that take place as a result of our environment. So things that we're exposed to, things that we choose to do, are placing what we call epigenetic tags on the DNA and on the histones, if you'll recall, or on the nucleosomes that the DNA is organized into. And that can all affect how those genes are expressed. And that's semi-permanent, meaning that those changes, those tags, can be passed on to children, to our children. And from not only to our children, but possibly to our grandchildren. So all of this is under investigation right now, but it's a whole new field of genetics. Epi means on top of, so or above. So this is above genetics. This is control of the expression of genes beyond just the simple sequence of nucleotides in that DNA. A very exciting area of research.